Hello and welcome to ICDD's InSession webinars.
Soon you will hear former ICDD Executive Director Tim Fawcett presenting pharmaceutical characterization using PDF-4 organics.
Please remember to visit icdd.com for information on future free webinars, educational events, product information, and to purchase ICDD's databases and software.
Okay, so today we're going to talk about pharmaceutical analysis using PDF-4 organics. And the data that you're going to see today comes from a study that we did of top prescription drugs in 2018. And we didn't do all the top 200 in the world. We did about 40 of the top 200 that we were able to get with the help of Mainline Health in Pennsylvania. We also analyzed some over-the-counter pharmaceuticals and some prescription drugs. And what you see is the particular brand name of the material that we saw, what its active pharmaceutical ingredient is, and in total, this was 65 different formulations that we analyzed. And one of the purposes of this study was to see if the ICDD had the correct reference. Now, when I define the correct reference, we're going to define this that it is the ambient determination and the specific polymorph that's found in the commercial product. So lots of databases will tell you that they have different active pharmaceutical ingredients, but since most pharmaceuticals have multiple polymorphs, we're looking for the ones that are found in the commercial product. And if you're in law enforcement or if you're looking at competitive analysis, this is the one you want to identify. The other thing I'll specify is ambient determinations. Most of the time, you're doing a room temperature analysis and you want the reference materials that you have to be ambient determinations so that you can go through the identification process successfully. Now, these studies were written in two publications that, like I said, were published in 2019. One was a practical guide to pharmaceutical analysis. The other one was formulation analysis of pharmaceutical drugs. We did analyze gel caps, pills, encapsulants, microspheres. There was a pretty wide range of both materials and material types in these 65 materials. And the reason why I mentioned these two papers specifically is this one's available on the ICDD website and you can download it for free. But just as important, it is a review article of pharmaceutical analysis and it has over 84 references that you can cross-reference. The formulation analysis one has over 44. So you really have all the reference links to what's been going on in pharmaceutical analysis for the past 20 years. And that's why these are a valuable resource. So what I'm going to talk about today is I'm going to give you a short introduction. We're going to talk a little bit about product design because we call this a targeted database. And I'm going to try to give you an idea of what we mean by a targeted database. And then we're going to talk about the different types of data in the database. If you know the design and all the different types of data that we put into the database, you can do effective data mining. This is a relational database with all sorts of Boolean operators and searches, and it's really pretty amazing what you can do from data mining. And then once we go through that, we're going to look at the ways to identify a material, and there's several different methods. And I'm going to cover these methods because we're going to actually be combining them in most of the analysis of pharmaceutical tablets through a process that we call whole pattern analysis, where we're using both search match and different types of pattern recognition programs to identify materials. So in the introduction, I wanted to bring out the point that pharmaceutical analyses are rather difficult. And that's because you're typically dealing with asymmetric crystals that are a low symmetry, particularly needles and platelets. The majority of your active pharmaceutical ingredients do form needles and platelets. And the diffraction is weak relative to other materials because you're working with low Z elements, carbon, hydrogen, nitrogen. What this means is the X-rays penetrate deep into the sample. And because they penetrate deep into the sample, you can get both transparency errors, where you get diffraction from different levels of your sample, and you get displacement errors. And these errors are frequent. Now, if you happen to work in inorganic materials or mineral analysis, geological materials, these type of things you may bump into 10% of the time. If you're working with a pharmaceutical, you're bumping into these two problems about 80% of the time. The third thing that you got to look out for is non-crystalline materials and nanomaterials. And these are part of the product design. And they're part of the product design because these materials have high surface area and they're involved in fast dissolution and controlled drug release. The controlled drug release is usually through hydrogen bonding of the materials and is a design element that people use in pharmaceutical formulations. Now, to handle these type of problems, your software system and your database has to have tools to handle orientation. And we're going to show you a March to Laissez function. If you use JADE, you also have a spherical harmonic function. You need to correct for common instrument errors. And I would say most of the software on the market can do this today, but you've got to know how to do it to get the transparency errors and displacement errors corrected. And you have to have a database that has non-crystalline references. So almost all the other databases for powder diffraction out there are derived from single crystals, and they're not going to handle the non-crystalline references. This is a really pretty important tool and a major advantage of using PDF-4 organics over almost anything else that's out there on the market. So I'm going to show you this and highlight this with the analysis of Benicar HCT. The red pattern here is the diffraction pattern, and then we have the references. We have a small crystallite size alpha lactose monohydrate that we've identified. I know it's small crystallite size because of all these clusters of peaks that are merged together, and they're merged together because the peaks are wider and they form merged peaks instead of single peaks. Then we have one of the APIs, olmesartan medoxamil, and that's in green. And we have another API, hydrochlorothiazide, and this shows another typical problem that you have with pharmaceuticals, which is strong peak overlap. If you look down below, the blue phase and the little tick marks are where peaks show up, and the green phase overlap with every single peak that's in the hydrochlorothiazide. So you're not going to find this phase unless you do a very, very good job in deconvoluting the spectra and finding out the residual, small residual intensity that's left over that enables you to identify this material. And then we have this baseline that seems to float up and down, and that's because this material has nanocrystalline cellulose and amorphous cellulose. Now, this example is also a good reason that we show that the PDF-4 database is continuously evolving. There's new materials coming onto the market all the time, and you have to be on the lookout for those. And when we started PDF-4 Organics in 2002, it had 146,000 entries. And now it has 560,000 entries. And in that last example that I just showed you, we didn't have the crystal structure of cellulose 1B until 2010, and that's because that was a special project by Jim Kadick, where he did an amazing job elucidating the structure using a fiber pattern, solid state NMR, and synchrotron X-ray diffraction pattern. We determined amorphous cellulose in a special project that ICDD had with International Paper and Eva Bucher in 2010 and 2016. We had the pattern of hydrochlorothiazide. This came from a single crystal structure from A. Florence and all in the UK, and ICD editors contacted him and was able to get the powder data supplied to the ICDD. And then the pattern of olmesartan medoxamil came from a special grant by Xiaolong Chen in China and from the Wuhan Biocar Biology Technology Company. He was able to get a sample from them, and we know that that particular reference that he got is what's matching Benicar HCT here. So this has been a continuous evolution. And if we go back to our 2002 database, we only had this material, and we've subsequently added these as targeted materials. We went out, we didn't have these materials in the database. We found these materials through different routes and added them to the database. So that's what we mean by a targeted database. Now I'm going to talk a little bit more about the product design. Organics have always been part of the powder diffraction file, even since the 1930s and the original 150 patterns that came from the Dow Chemical Company. And there's been a long-standing group of people that have been ICDD members that work in the pharmaceutical industry that are part of the Organics and Pharmaceutical Subcommittee. And they helped get data in to the database from those early years until we had 24,000 entries in 2001. But in 2001, this group came back and said, we need more data, we need more targeted data. 24,000 entries isn't going to do the job. So then we had a dialogue with that subcommittee, and we had a bunch of very interesting conversations that also helped shape the product. They said, gee, we're looking at pharmaceuticals in a plant. We may be looking at a pipe clog. We may be looking at a filtered residue. So we added common phases to this database that included all the common corrosion products. And these were primarily inorganics and metals and metal oxides. We added all the common salts because when they're looking at raw materials, they could have salt as a byproduct. So we wanted to distinguish the salts and the API. They added the entire polymer subfile because some polymers are used in pharmaceuticals in the formulation itself, but there's other polymers, there's different polymers that are used in the packaging. So in order to analyze both the drug and the packaging that was in, we added all the polymers that we had into this database. And there was a need to get all the common excipients. And these could be inorganic, organic polymers, whatever a pharmaceutical scientist uses, we wanted to put those materials into the database. And to understand what we needed to get, we were directed towards the US Pharmacopoeia, European Pharmacopoeia, and the excipients handbooks. So lists were generated. We checked those lists of what was in the database, what wasn't in the database. And then special projects were initiated to get the materials that weren't in the database, to obtain them and get reference materials from them. And finally, the pharmaceutical scientists said an awful lot of the powder patterns are part of patents. And usually the patents only have a partial description and the quality is very low. So we had a special team of editors extract the patterns, process them, try to get as complete description as they could, calibrate the data, and then enter them in data in the database. Then we used our strategic alliances. So ICDD had strategic alliances with ICSD and NIST already. And this particular product really highlighted the strategic alliance with the Cambridge Structural Database and their large collection of organic patterns that were collected from single crystals. And this is how the targeted subfiles were generated. So we took all the data from the different sources, and then we developed subfiles dependent on function. So for the pharmaceutical industry, we wanted to have the best subfile for bioactives, pharmaceuticals, amino acids, and polypeptides. Now, we also targeted the chemical industry and law enforcement with this product. So we also have forensics, explosives, carbohydrates, polymers, and pigments. So these are the industries targeted. Now, if you have an organic material that shows no bioactivity, no pharmaceutical activity, and isn't in one of these special subfiles, chances are it doesn't have a function and it's not in the database. But we use all those alliances to get everything that does have one of these functions and put it into the database. So the contributions from the different groups aren't equal. There's a large part from CSD, but we were very selective on the parts from the inorganic world and the polymer world and some of the polymers and non-crystalline materials that were donated. Now, if you take a look at the pharmaceutical phases in the pharmaceutical file, you kind of get an amazing result. And then you have to look at it because what it says is actually the patterns from the ICDD are the largest contribution into this subfile. And that's not an accident. There's been over 1,800 that were entered through ICDD grants where we targeted materials and got them. There was another 20 from special projects. These were mostly non-crystalline materials. There was another 992 that came from private donations and communications with these companies over here. You can see the US FDA, Eli Lilly, and University of London as highlighted because they supplied many, many patents. but we also got ones from Japan, from Scotland, from Finland, from India, all around the world. And there's over 1,000 of these that were private donations into the database. And so these are the things that you don't see in other databases. And it's about half of the total powder diffraction reference materials is from these targeted additions. And then we have the ones from CSD and from the other sources. Now, when you go to bioactives, the story's a little different. Here is where the CSD shines. They have lots of bioactives, and there's smaller contributions from the other sources from ICDD. And these are things like depressants, narcotics, pesticides, antimicrobials. It also includes other antis in here. If it's an antifungal activity, bacterial or parasitic, it's in this bioactive subfile. It also includes materials that may have been tested and tried for bioactivity, like over 1,800 anti-cancer agents that never made it to a commercial product. If it made it to a commercial product, it's actually in the pharmaceutical subfile. And you can see the high number of patents that are also here. That's an indication that it went out into the marketplace. Here we have 13 patents, but we have a lot of anti-cancer bioactives that never went to the commercial activity. So these are also not drugs that weren't approved, the drug derivatives or metabolites, anything that showed bioactive activity. Now, the reason why I point all this out is if you're in early stage research, you want to use the broader classifications of bioactive common phases and pharmaceuticals. If you're looking at a commercial product, you want to go directly to the pharmaceutical subfile. And does that mean that really 13,000 phases are more important than the other 560 that are in 1,000 that are in the database? Absolutely right. We created these subfiles so when you do an analysis, you only get what you need and you eliminate all these other false positives that don't have the right chemistry. If something's in the pharmaceutical, excipient, narcotics, or psychotropic subclasses, that means they are defined and they're controlled. And these are the groups that create the definitions. So they're defined, they're known, you find them here. And if you're working with a commercial product, you go right to the right subfile. Like I said, if you're at earlier stage, you might want to use pharmaceuticals, common phases, and bioactives. Or if you're looking at something like a pesticide, you go right to the bioactives. And this is 45,000 entries of targeted materials. So now that we've talked about some of the functional materials in the database, I'm going to talk to you a little bit about database types. We have several types. We do have over 30 subfiles, but these are the key ones that we've talked about already for this particular product for the pharmaceutical industry. We also have data that are different types by their crystallinity and by their form and format. So we have patterns calculated from single crystals. We have patterns calculated from disordered single crystals. These are much more challenging because you have to look at where they calculated right. Did you get the electron density in the right location? This all goes into the accuracy of the pattern that you calculate for it. We have patterns calculated from older data and DI list, and a lot of statistical analysis goes in to make sure that our patterns calculated do represent the materials from this older data. We have new data that's from three-dimensional periodic crystal structures, from dimensional crystals that only have two-dimensional periodicity, like some clays, from nanocrystals and amorphous materials. And all these are reviewed and classified by the editors. They're all studied. And you'll find the editor's comments embedded in the quality marks and in a special comment that goes with each entry that's called the editor's comments. So I want to point out, this is the intelligence that's put into the database. It's more than just data. It's subfiles by function. It's review of the quality of the material and any other unusual features by the editors and the quality marks. So these are the things you want to be looking for when you look at the data. And these are the people that make it happen. So when we talk about using artificial intelligence, using intelligent algorithms with the PDF-4 organics database, it is based ultimately on people. Their intelligence goes into the database, gets coded, and then it's available to you. So Amy Gindhart was doing and her team have done the wonderful job of looking at patents, extracting the data, figuring out how to digitize the data and how to calibrate it. Stacy and Amy have worked on these lists of excipients and new materials. I'm going to shout out a highlight to Jim Kadick, who's a special member who's worked as a special project investigator. He's done more pharmaceuticals from powder to fraction, more structures than any human being I know. And he's got 150 APIs that have put into the database. Suri acts as the fixer in chief. He looks at all the classifications, all the comments, and if you find something wrong, you contact Suri and he fixes it. And he also figures out how to identify and fix things on a massive scale himself. We have Tom Blanton. He's our executive director and chief scientist, but in his role of chief scientist, he's an expert in nanomaterials and polymers, and he's made sure that we have the right materials in the right form and format into the database. And finally, I'll mention Justin Blanton. Most of the software we're going to be showing was created by Justin with input from all the members and other things, but him and his team have done a wonderful job of taking ideas and making it a reality through the software. We also have a lot of input from the PPXRD Symposium series, which was developed in the same time frame as PDF-4 Organics. And this is a symposium that brings together lead researchers, development scientists, and industry consultants together to meet about once a year. It's 16 over 20 years. But here's some of the people. And basically, when we get this group together, we listen. They're doing the most advanced things in amorphous material, pharmaceutical characterization. We listen, try to get their ideas, and try to embed it to the database. I'd also like to point a shout out to Dr. Ann Newman. She's also part of this team. She's an instructor extraordinaire and has also been helping out with the pharmaceutical PPXRD symposium. So all those ideas get into the database. Now, before we go and I'll show you the database, which is the next step, I do want to point out ambient and non-ambient. Many of the single crystal structures of organic data sets between 40 and 50% are collected at low temperature and non-ambient conditions. What this does is it shrinks their despacing values to low value. And this is why we put in a special check for ambient data. So if you're running a room temperature analysis, which most of us are doing when we're looking at pharmaceuticals, you want to check this box so that you get the right non-ambient determination. And this is part of having the right things that are the right part of the analysis with the right polymorph. Because once you shrink these despacings, sometimes you get possibility for a phase change, but a lot of times you'll actually miss the identification because the critical despacings and unit cell parameters are all at lower values. If you don't find what you're looking for with a search through the ambient, then you can go to the non-ambient, but you're going to need to go into your software and open up your search windows because the materials aren't going to be at the position that the room temperature materials are going to be at. So this check mark has a very practical basis when you go in and try to analyze and study different formulations. So with that, I got a little reminder that now's the time that I'm going to go to the database and show you the database live. So we're going to go here. And you should be seeing the database now. And here's the opening screen of the database. And I'm going to blow up the opening screen, but you'll see several of your different types of modules here. And we're going to be using Civ later on. That's gives you the ability to search, match, and identify materials. But here's our subfiles. So we talked about bioactive, which is right here, all the subclasses. So you can search as a whole, or you can look specifically for narcotics, stimulants, psychotropic, pesticides. You can see the subcategories. We also have the pharmaceutical one right here. And once again, you can look at it as both APIs and excipients, or you can separate out the excipients. We talked about the ambient, non-ambient, and recommending that most of the time you search on ambient. We also have the status, and we have the quality marks that we also discussed up here. Now, I'm not going to get into the quality marks in detail. If you want details, when you use the database, you can either look at the help file, or if you hover over some of these quality marks, sometimes they'll explain what they are. But I want to basically say, when you use the quality marks, we tried to make it simple. There is green, means you have high quality data. We didn't find any errors. If it's red, it means there's an error in it, and you should probably check the editor comments. Here's one of the comments that defines what low precision means that just popped in. If you're in the yellow, it means that we found a minor problem, and you should check with the editor comments to see if that minor problem interferes with the particular thing that you're looking for in the analysis. But green is go, red means you have to be a little bit cautious, yellow says that you should probably check and see what the error is and see if it affects your results. Then most of us have some sort of elemental analysis, and this is just an assist that if you know some of the elements, you can put them in. You don't have to, but you can have that as part of your search. Probably the tab that I use the most, and you can see we have a lot of different types of searches. This is part of the artificial intelligence in the database. You can search and cross-correlate all sorts of parameters. But the thing that I use most when I'm doing pharmaceutical analysis is the name. And that's because most of the time when you're looking at a drug, you already have an idea what the chemistry is. You're looking at a specific problem, a specific formulation. So you can either do a general search and a search match, or you can do something like put in a word or part of a word, and I'm going to put in fexo because I'm going to be searching for fexofenadine, a common antihistamine. But if we drop down here, we can look at the whole name or a mineral name or an alternate name, or we can do part of a name or part of a phrase or do it exactly. So since I have trouble with very long names, I usually look at a small fragment. I hit search. And then we get what we call is the results table. So it's going through the entire database. It's looking for anything that says fexo. And we're going to get a series of fexofenadine patterns. So these are going to load up. It's taking a minute. There we go. So this loads up. And when we loaded this up, we have a number of fields that are available to us. And this happens to be a series of fields that I like when I'm looking at a material like fexofenadine. And right away, from looking at the series of materials, and I'm just scrolling through the different materials, you can see from the two-dimensional structure that they have slightly different structures. Some have the same structure, but some are different. You can see from using in the alternate name, that's where we put in trade names. So you can see there's fexofenadine, Allegra, Telfast, the Allegra T, so venlafaxine. So you have the different derivatives. You can also see there's a large number of salts. Since this was one of the first anti-drowsy antihistamines on the market, it wasn't the first, but it was one of the first. It has lots of derivatives and people have looked at lots of applications. So we have over 18 materials. So then the question comes, what's in your sample? But before doing that, I wanted to show you some features of the search field. One of the things you can do is you can put, once you get a series of displays, and display was all based on fexofenadine, you can add and subtract any fields that you want. And you do it through this fields icon. And here's just some of the fields you can look at. I showed you some of the ones I'm looking at, but if you want to look at all the crystallography, here it is. All the diffraction peak locations, here they are. Wide range of physical properties, including some type topologies, different author information, different comments. Now, the ones I chose were things that I find to be useful, but maybe you find other ones to be useful. I also like looking at the database comments because that's where they'll usually talk about polymorphism. And I like to look at the authors. And sometimes this WO means it's a world patent. So it's telling me that it extracted this information from a patent, which to me is useful to know. So then we've looked at all these and we may want to sit there and say, okay, well, I have my drug. How do I know it's one of these? Well, even right here in the search file field, we have a tool, and this is a similarity index, and this is a pattern search match system that's just based on pattern recognition. So it's taking thousands of points that you have in your reference pattern, looking at thousands of points in the reference and comparing it one-to-one. So this is a type of search because it's using lots of data that you don't want to do on all the database, but if you have a smaller subset of 18, or even something like 13,000 that you have in pharmaceuticals, this can be a very effective search to answer the quick question of, do you have this in your material? So I'm going to look at a scan that I took of Allerfix. And we're going to see the scan here, and I'm going to simply import it. And then down here, it's running through the 18. It's comparing every one point to point, and you can tell at a glance that this material is not in your product. This material is not in your product. It's not matching. This polymorph doesn't match. So we have 3 forms of fexofenadine and different salts that do not match. And in this one, you'd look at and say, it has a couple of peaks, but maybe not. But even here, we can open this in a larger window. We know in this particular material that the active pharmaceutical ingredient is actually only about 10% of the pattern. So I'm going to go to pattern settings, and these are just all options you can use. I'm going to scale this pattern down to 10%. I'm going to apply it. And then I say, well, we've got the 2 peaks here, a major peak here, a major peak here, which would mean that this might be a candidate for what's in this particular drug. Now, I would never do it based on this quick analysis, but this quick analysis could say, of those 18 things I was looking at, which one is the most important? Which is the one that I should focus on? And it's telling me this is the one I should focus on. Now, the reason why I'd say I'd never analyze it, I'd want to do the complete analysis first. These peaks here is a cluster that's of alpha lactose monohydrate. We have broad profiles here, which is going to turn out to be cellulose. I know this because I did all 65 of those drugs, and I know what those patterns look like. And then if you analyze the alpha lactose monohydrate and you look at the cellulose, you'll see that the peaks you haven't identified tend to be these peaks that all belong to this particular polymorph. But you can take a quick look and look right away by doing that type of search that I just looked. You can also look at this hydrochloride versus another hydrochloride. This is also an effective way. We could highlight 2 or 3 of these materials at the same time. We can go over here, simulate the patterns. And if I blew this up and then offset all these plots, what this tells you at a glance is these are not the same polymorph. You have different polymorphs, different salts. These are not identical. Sometimes, by the way, you'd find out they were identical. And then that's a critical piece of information that you know. So there's a lot of quick things you can do, quick searches you can do just through this search form and looking at the different properties. Sometimes I use this form if I've eliminated all the other materials and I only have a few strong lines left, I can do a search of just the strong lines. And that's also something that I've frequently done with pharmaceuticals. So there's lots of power in the data mining. I'm going to go back to the presentation. Okay. And then we're going to talk about material identification. Now, if you're identifying materials, there's the classical way where you use the peak locations and their intensity. And here's the peak and the location of Eliquis. And then you match it up to your reference materials. So I've matched it to cellulose, to lactose, and apixaban. And here we say, okay, here's all our unknown peaks, and here's how we're matching it to these different phases. And we get good matches across the board. And when we have a particular good match, it puts it in the color blue. Now, I chose this example for one thing. You see most of the materials are in the color black, which means the match is not perfect. And that's because you have overlapping peaks. This one observed peak actually has 2 contributions. This peak has 2 contributions. This peak has 3 contributions. So you're working with merged peaks, something that you see very, very frequently. So your observed peak is slightly different from the reference because you have peak merger. So this is something to look out for. But this is the classic way that most of your software works. It's looking at the DI pairs, matching up the patterns. And no matter whose software system you've got, this is the old classical system that's been used for years. Then we've already showed you some of the pattern recognition. Here's an Allegra shell. I ran it through the pattern recognition program that's called the similarity index that says, oh, I see that the general shape goes up and then goes down. Cellulose goes up and goes down. Turns out that cellulose is in this particular material. Sometimes this sort of pattern recognition program may be called integral index. And in the similarity index or integral index, we're usually comparing one experimental pattern to maybe 1,000 references. A cluster analysis uses some of the same algorithms, but that's usually doing a matrix where you might have 10 patterns and it's comparing it to 10 other patterns, all in a 10 by 10 matrix. And so cluster analysis, by the way, is in JADE. The similarity index is in PDF-4 organics. And we use the PDF-4 organics mostly to look at nanomaterials and amorphous materials. The similarity index works perfectly fine with crystalline materials, but it really does exceptionally well where you have broad profiles and it says, gee, do we have the same general broad profile? So here's one type of tool. Here's another type of tool to identify materials. These are just variations of the classic tool where it's putting emphasis on different types of data. Hanawalt's the classic where it's putting it on the strongest lines. Here it's putting it on the what's called the long lines, which are your high D spacings, which can be useful in pharmaceutical analysis, just puts emphasis on it. Sometimes it'll help you identify more than other ones. So when we search for pharmaceutical materials, we're going to use the program Sieve. I'm going to show you this live as well. Here's a sample Seroquel. And if I search it, we're going to identify through the similarity index. So here I can put in a similarity index and I can put in the goodness of merit, which is based on D spacings. And here's your D spacing matches. So here's one way of identifying. Here's another way of identifying. You can do them both at this program, or you can use those other options to run them separately. But it's matching up the second highest peak. It says we think we like cellulose. By the way, the first one, it says we think it matches polyethylene glycol. That's not a good match. Polyethylene glycol will account for a big peak here, but we know that's something else. That's actually the alpha lactose monohydrate, which I show down here, which has almost a perfect match for all its D spacings. So this is where you actually have to use your brain. The software is telling you you have a couple of candidates, but then you have to go and look at the match, see how well it fits. Here you would pick the second best over here, and you'd pick the top GOM, which is the alpha lactose monohydrate, to get your answer. So this is how we combine both of them. And I'm going to show you some examples. The examples all use graphics produced by PDF-4 Organics. I did update some from some of the published data, and we'll talk about those. Everything was taken from ground powders. So this could all be reproduced from the product, all taken on ground powders. So I'm going to show you Prilosec OTC. Basically, we use the similarity index to identify the cellulose, and then we use the conventional search match to identify the API, which is omeprazole. And so this is what we call an offset plot, where the red is your experimental and your two references are shown below it. It's just a different way of graphics. And so some of these examples, I'm showing you different graphics, and it's always good to have programs with gives you very graphic options so you can physically see the fit, and what the analysis does and what your eyeball sees should be the same thing. So here we have Invokana, and here I have offset some of the excipients, which we call a stacked plot. So we have lactose, we have cellulose, once again, and we have an amorphous cellulose in the pink here. Now, when you analyze those three, we can also do a summation plot, and you'll see that there's missing peaks. And in this case that we published in 2019, we didn't have a reference pattern. So I used these peak locations. I looked for Invokana on the internet, just using Google, found this Chinese patent that's described form 1. And I said, oh, it looks like it's the form 1. Now, I fed that back to the ICDD. ICDD actually extracted this pattern, and I was right. And here's the form 1 in green that's gotten extracted from the Chinese patent. But in the meantime, a crystal structure was done. And the reason why I show you this example is is there's more like this in the database. Patents tend to give you a minimum amount of information. So they gave you the major peaks, but not the minor peaks. The crystal structure, we get the total information. So having the crystal structure was an improvement in this case. It is the same phase. We can do that by looking at the overlay. And now here's the fit with the summation plot, which is actually terrific. And then we have a new feature this year where we can set any material as an internal standard. And we happen to know that the canaflosin is 48% in this particular pill. So I could set that concentration here by locking it in. And then it calculates out the beta lactose and the cellulose with an RIR. So I get a quantitation plus a phase identification at the same time. Like I said, that's a brand new feature that we put in this year. The feature is locking in a internal standard. Here's the Benacar I showed you before, and I'm just showing you the summation plot, how you can sum all those different materials into a pattern. And here's one that we did. We wanted to show the versatility of the database. So we took synchrotron radiation from Singular. We had an amorphous reference of sodium monoleucas. It also contains microcrystalline cellulose. I blow these two up because what we're actually analyzing is the green pattern and the black pattern. And we did a standard addition experiment where we put in a little bit of sodium monoleucas into the sample. And so the original sample is in green, plus 5% sodium monoleucas is in black. That standard addition could go back calculate that the API was at 4% in the pill. It's actually in there by the formulation at 5%. So it did a very good job with an amorphous material with synchrotron radiation. We can do similar things with all our references for time of flight neutron and for constant wavelength neutron. So you can also collect data at any of the worldwide sources that have user programs, and most neutron and synchrotron sources do have user programs. Collect your data there, analyze it with the database, or analyze your lab data, and the database converts this back and forth to the different types of sources. And that's what we were trying to show there. Seroquel, similar. We have now two different excipients. We have both cellulose and hydroxypropyl cellulose, which is the green. Here's your API. Here's our alpha lactose monohydrate again. And then we had a stearate, I think. And then you sum those up. And once again, you've matched the entire pattern. So we were using our graphics tools. And I'm going to go live now, but here's another case with Uloric, and we match that up with Febostat. So here's a case where we have 6 entries in the database, but we didn't have the key one, which is this form 1 in a patent. I am told that's in process right now, and we'll probably have that in the next release. So when you quantify things, there is some difficulties. A large amount of data has come from special projects, has come from donations. So we only have 59% of the database with atomic coordinates if you do Rietveld refinements. And we only have 62 that have I over IC if we try to do the reference intensity ratio. So this is a case where it's nice, if you have it, to have a method that can analyze structures without atomic coordinates. And this emphasizes the importance of using standards and using things like internal standards, which is why what we try to do with the improvement this year, we'll put in the internal standard that can calibrate the material. So I just wanted to point that out. To go to the phase quantitation steps, you usually have to do a little bit extra. And now we're going to go back to the database. Whoop, I got to go share, and we'll go here. And now I'm going to use the program Sieve, and this is the program that identifies materials. So we're going to import a diffraction pattern. I'm going to go to my library of materials that I've analyzed. We're going to look at Xarelto. Now I can use some defaults, and I'll show you how the peaks are set up. I'm not going to spend much time, but I can show you that you do have choices how you set up the background. I like a linear background so I can analyze the nanocrystalline and amorphous materials. So we're going to remove the background, then skip smoothing. I strip out alpha 2, and I accept the peaks, and I see an extra peak here. You can just add it. We're going to import this, and we're going to do what I mentioned before. We're going to look at a pharmaceutical, look at only ambient, and I'm going to apply that as I filter. And you can see how fast the analysis is. Right away, it's coming up with rivaroxaban, and that is the active ingredient in Xarelto. And it's giving me two choices. There's two different crystal structures. You can see they're pretty similar. My third choice, and by the way, when I say a choice, this is based on goodness of merit. It's based on peak matching. And here's all the peaks that it's matching. So it's matching all the major peaks in the pattern. They're all in red. That means it has a good match. If I want to look at all the peaks in the pattern, here's all the peaks in the pattern, matching with the API. Here's my experimental. Here's my match peaks. You can see how many of them are in blue. It says it's a good match. And then my other choice is alpha lactose monohydrate, and you can see that matches well too. Now, I can also look at the similarity index. And if I look at the similarity index, I get a different list. And now it's just doing pattern recognition point by point. And you can see that it matches up with the alpha lactose monohydrate. So if I'm trying to identify the ingredients, I can stop here. I know I have the alpha lactose, not the alpha lactose, I have cellulose 1 beta. And then if I look at the GOM type analysis, I have the API and I have lactose monohydrate. So those are my three phases. Now, the reason why I wanted to show this to you at this time is once I start analyzing phases, the cellulose goes away. And it took me a long time to figure out why it was doing that. And that's because the API has its major peak right here. And if I look at the cellulose reference, so we're going to look at the similarity index. Okay, we're going to look at the cellulose. It has the same major peak right here. So if I start crossing out the API, it starts wiping out the same peak that the cellulose uses. So it took me, like I said, several tries before I figured that out. So I'm going to look and find out what this PDF is here. It's 601502. That's the reference. I'm going to go back to the GOM. I'm going to add in the API. We're going to add in this lactose hydrate. I'm going to accept that phase. And you don't see the cellulose anymore because it's already accounted for a peak here. So I'm going to show you a trick. You can insert a material, and we're going to insert the pattern 000601502. This is also useful if you know you're working on a drug and you know it's at low concentration. Sometimes it's hard to nail it on the search match. You can actually put in the candidate, do a search for the API, put in the candidate, and add it to the matches. And so now it's added cellulose to the matches. We accept that. And we've got our three phases. Now in this particular case, I want to do a little bit more. So we're going to adjust this. And we're using a different module. Okay. So what I mean by do a little bit more, I wanted to do things like maybe look at the crystallite size, maybe look at the quantitation. So the first thing I see when I'm looking at my three phases and my identification is my cellulose is a little too high. So I'm going to adjust this down to 0.6. Okay. And we'll update it. And now, because I'm close to having all my peaks, I can do a least squares fit. If you're not close, sometimes it can refine away and give you a bad refinement, but here it zeroed it in. I can now check my summation plot. And you can see now it's summed all the plots versus my experimental pattern, and it looks pretty darn good, but it's not perfect. So we're going to say, okay, and it's not perfect if we blow up a region of it, because you can see the peak widths are much sharper than what you have in the experiment. And that's because you have crystallite size differences. So we're going to look at the crystallite size of the alpha lactose monohydrate. And so you have a a panel over here where you can change sizes, you can look at orientation, and we're going to look at smaller crystallites. And I'm looking particular in here where these peaks merge, and that's not too bad. I'm going to take a look a little bit more. I like right around in here. I'm going to do a similar exercise with the API. Okay, and I'm going to change the crystallite size here. Here we go. And you can see once we change the crystallite size, my intensities aren't quite on the nose again, so I'm going to do my least squares fit. And then it looks like to me my cellulose is a little low, so I'm going to put it up to 0.4. And now you're starting to see that we're getting a pretty darn good pattern match. I can then go and reset the view. I have options of lots of graphics, so the other thing I can look is the difference, and I can scale this to the total point. And this is showing me maybe a little high here, a little off. By the way, a little high here, that could be the orientation. So I can also go to the alpha lactose. I can look at the major peak 110. I can start adjusting the peak intensities. And once again, I get that to a point where I want to fit, which is just a tiny bit of orientation leaning towards a needle morphology. And so now I have a whole bunch more information. I know the crystallite size of my components, 325, 260. I know the orientation. I've got a summation that matches everything, and I'm feeling pretty comfortable. Now, I also know that this particular sample has 22% of the active ingredient. So if I want to finish this off, I can use this as an internal standard. I can do OK here, and it'll calibrate and give me a quantitation. Oh, I didn't lock this, so I can do 22% here. I lock that phase. Now it calculates everything to the 22%. And it says I have about 30% left over. And that 30% is in the cellulose. The cellulose calculation is a little bit off because we have a nanocrystal here, and it's looking at the ratioing, and it's actually a higher volume. So now we have, about 30% cellulose, 22% the active ingredient, and 45% lactose hydrate. And with that, I'm going to stop and hope you enjoyed how you can identify things, look at the quantitation, and the key functions of PDF-4 organics. Thank you.
Great, thanks, Tim. So we have a few minutes left, and we have a few questions from some of the attendees. So the first question is, could you show us how to list the pharmaceutical materials in the organics database that have raw diffraction data?
Okay. That's actually pretty simple. Is my screen still showing?
Yes.
Okay. So I'm going to go back to the search. Oh, I got to say okay here. I'm going to go back to the search screen. Okay. And thank you for asking that question, because I meant to show it and I didn't. You have two important keys here. If you're going to do a follow-up Rietveld, you might want to hit atomic coordinates, and then it's only going to search on things with atomic coordinates. Now, I showed you in the pharmaceutical analysis, that's usually very helpful when I'm looking at minerals or metals, but in a pharmaceutical analysis, not everything has atomic coordinates. Now, the other thing is you can look and search for raw diffraction data. And this is where we have all the amorphous materials, the nanocrystalline materials. For the last 20 years, we've been asking people that characterize these materials to send us their raw data, because you generally have broad profiles, not sharp profiles, and the raw data files are much more telling in whole pattern analysis than a despacing is. So this is where you find them, raw diffraction data. So you search, you click on this, you hit raw diffraction data. So if I had a pharmaceutical and I was looking at one of these amorphous materials and wanted to figure out if it was in my pattern, almost like when I ran the API, I would run pharmaceutical, raw diffraction data, I'd run a search, and I get the searches. It's going to come
You
up
still
right
have the
here.
name search selected.
Up. Thank you. So first of all, you want to clear all your tabs. By the way, the database is always right, and Justin was right. It told me that I did the wrong search. Raw diffraction, pharmaceutical, search. So now it's coming up with all the things that have full patterns. Now some are crystalline, but it will include all the amorphous materials. And then if we wanted to do a search, we could do that same similarity, input our data, it would compare it versus all the other materials. And this is now searching 1,070 materials.
All right, great, Tim. So we have another question, and it is, can we analyze biomolecules as a surface modifier on nanoparticles?
Typically not, because I'm making the assumption in this case that the molecules are only a couple of atoms thick. Now, that may not be the right assumption. So I use the word typically. There's people doing amazing things like that using synchrotron data, where you've increased your sensitivity, increased your resolution. And I've seen people with synchrotron data look at layers that are 1 or 2 molecules thick. So as long as they're there, you can do it. I've also seen some amazing work done by a person called Simon Bates. He is a pharmaceutical expert, but he's actually analyzed the scatter of water on the surface of pharmaceuticals. And water has its own diffraction pattern, and he's been able to look at 0.15% water on the surface of pharmaceuticals using high quality lab data or synchrotron data. So you can do some pretty amazing things, but that takes usually takes some extra effort in the data collection. You got to get really high quality data to look at small differences like that.
Great, thanks, Tim. Well, we're at noon U.S. Standard Time, so I think that's all the time we have for questions.
Thank you, everybody.
Don't forget to like and follow ICDD on Twitter, Instagram, LinkedIn, and Facebook. More information on ICDD.com. Contact us at info@icdd.com.